With that, we're going to take a little bite into lunch. We're running a little over, but I know we're super passionate about rigor and reproducibility, so I know nobody minds. And I'm going to hand it over to Rui to lead that session.
Well, thank you. I'd like to start by thanking our speakers. I don't think we need to introduce all the speakers. They've been introduced, but I do want to thank them for the opportunity of having them here in this panel. I was not only surprised today with the wishes of happy birthday, but I was also surprised in the coffee break that I should lead this panel discussion. I'll try to do my best with the questions I came up with. They're not very rigor, so I don't know if they'll be very reproducible, but let's hope at least we have some kind of discussion here. We've some of you have defined rigor and reproducibility, and I think here in the context we can. I loved your definition, Travis, and I think I completely agree with you. Reproducibility comes way before, and the rigor also comes way before. But I guess we can think here in this context of rigor or the lack of rigor affects reproducibility, right? So lack of rigor consequently drives lack of reproducibility. So in this context, and I'll ask everyone in the panel, including Dr. Novo, I just wanted to say Dr. Novo again. What are the sources of lack of rigor and reproducibility that you most commonly see in your own experience, in your own field, in your own technologies, etc.? So what are the major sources of lack of rigor and reproducibility? Anyone wants to start?
I guess I'll start because I'm over on the left side of the table. I think that one of the things that I run into a lot is time is a big one, right? So especially in the in the corporate environment, I'm pressed for timelines all the time, and you get to a point where you need to produce data in a short amount of time, and you get kind of painted into a corner, and you just kind of shoot from the hip and go for it, and that's when it bites you. So for me, I like to, especially when I get new people, I have them go through really basic experiments and do them 4 times and have them realize that stuff happens that we can't explain, and usually that vets itself out. If you do something 10 times, you'll know the truth. But if you do it twice, you won't.
Right.
And so the rigor comes from reproducibility itself and implementing that on a daily practice and demonstrating it to people early in their career, and the nuances that can change the outcome, whether it's not staining 45 minutes after you isolated your cells versus staining 5 minutes after you isolated your cells, and having those practice just become ingrained in them, that nuance can really come and play a role in your results.
Right. So you would say also assay optimization, right? Having time to do that, right? Good point.
And sort of on that note, I would say that either willfully or accidentally, a lot of times that stuff's not communicated well. And so I even recently we tried to reproduce an assay and followed all the protocol, and there was like a secret step that the staining protocol had to go in a very specific order, and we didn't know, and it didn't say anything. And I contacted the authors, and they were like, "Oh yeah, it was just that's the way we do it." And it turns out after they published, they realized it doesn't work in any other way. And so, and on a subject near and dear to my heart of unmixing, for example, there are multiple instrument vendors who do a custom form of unmixing that they don't report how they do it. And they tweak the algorithm in various ways, making sure, of course, they register the trademark and register all the PR and buzzwords, but you don't actually know what they're doing. So you literally can't reproduce the math or the work that they're doing at all. And this is not obscure vendors. Sony does this, BD does this, and you literally cannot reproduce the work that they do. So that also something that has to be clarified, is documenting properly what you're doing and transparently.
So I would say that in the lab, I think the biggest problem, the barrier to rigor that we have is planning, right? So like you were saying, like timing is important sometimes. So planning what you're about to do first before you even set out to do it, but also planning before you set out to do it. So, don't start your experiment at 11 when you're hungry before lunch, right? Or make sure that the protein that you need to coat the wells is thawed out or check that it is, because if you only have a small amount, now I have to wait to thaw it out. And of course I grabbed it right when I needed it. So like if you make sure you have everything together, because it's not stimulating your cells exactly at the right time, will have big effects. So planning, I think, is really important. And then from the microscopy or the core perspective, what we see a lot is that people kind of either we have two groups. One group that just thinks that the instruments are always constantly changing so much that you can never do anything. And then that's not the case. I say they're pretty stable, thankfully. But then there's the other group that those people that think that always changes, they don't concern me so much, because they actually try to keep track of that, even though it's not probably necessary. But the ones that think it's solely stable every day, and every day you do something will be exactly the same, that's not the case either, right? So it's more towards being stable. So what happens is they don't have the right controls, right? And these aren't even experimental controls, they're imaging controls. So that way I can compare my data set from day to day with other days, right? So and then make sure that everything is set up correctly, use the previous settings as a starting point. So really understanding that you need to have the right controls for the instrument, the right controls for your experiment, and that you can't do them without it, I think is a real problem.
Pretty much a broken record. I was going to say reference controls. The common theme that's cropped up here is people coming in with just their multicolored sample and nothing there. And also not knowing what they're doing, which is why I try to get people to consult the core beforehand. Because I've had people show up and say, here's my sample, Travis, it's orange. And I go, I can get an orange color out of all 5 lasers. Which orange are we talking about? And you just have to send them back to the lab to get that information. Or they come down to do a sort with a dry tube. It's education.
Right.
I guess it's my turn. So pretty regularly, like we have a series of slides that we sometimes present on this very topic about, the image stream. Obviously been in the imaging flow world for a long time now, but the whole idea is to keep an open mind, because there will be people who come and run something on a new piece of equipment. And when they learn something new that doesn't fit with what they thought was happening, it's immediately disregarded. And they'll say, this isn't the way that it looked on the Aurora. And I'll say, you have no idea how it looked on the Aurora because you don't have pictures, right? Like you learn more information. And yeah, it's not, the data isn't going to be exactly the same. It's not going to look exactly, because it's a different instrument, different detection. And I'm, as we have more imaging flow coming in here and we're getting to see more and more of what's going on and we're seeing new things, different patterns, it makes it so that as a scientist, instead of just saying, I'm just going to forget about that part, to then re-engage and be able to do that rigor and that reproducibility, because you now have more information about what's going on, instead of just saying, I'm just going to not worry about that part of it. And then using the algorithms to say, how can I apply this, instead of just what did grad student number 1 count, what did grad student number 2 count, and what did grad student number 3 count, so that it becomes applicable, so you can batch process, you can then apply it across multiple files. It just becomes really important to keep that open mind and use it as use the other technologies, triangulate your position so that you have multiple methods that are showing maybe not always the same thing, but if they don't show the same thing, then why? What's different about it? What is it that you're going to learn? So,
yeah, thank you. So I actually have I had a question for David, and since I'm the moderator, and it's my birthday, so I think I'm gonna ask this question, and I'm gonna ask the question to I'll ask the question to everyone, because this is something also a discussion dear to my heart. So you did mention a lot of examples, and Travis, you also did, examples of manipulating data in imaging, Western blots. You even showed some flow cytometry plots where people have manipulated those plots, repeating them, etc. But one of the major sources of data manipulation in flow cytometry is tweaking compensation or tweaking unmixing after the fact, right? So here we're trying to essentially manipulate or change the data according to what we expect it to be. And you mentioned, David, right, that this is something that's considered even fraud, right? Especially if it's done on purpose. So what are your thoughts here in terms of, again, this is one of the things that we most commonly do in flow cytometry. How do you think it affects overall rigor and reproducibility in flow cytometry? And this is a question for all of you.
Well, it definitely impacts it, but I mean, we'll try to do the best practices and try to get people not to do it.
Mm-hmm.
Now, you might flame me, but in my analysis class, I actually show people and do manual compensation on 2-color beads, but that's to show them the process,
Right.
not that that's what they're going to do.
Exactly.
Now, if I have a user where they come in with reference controls so badly gacked up that I have no choice but to still try to help them with their experiment, I at least try to tell them, this should not see a paper. But just so you have something to springboard and work from, we're going to do it this way. But again, I hope that the education in the core is people are going to do that. Now, here we go. I can also throw the question back at you. Once they walk out of the core, and I see that it's adequately comped, they take those files, they go back into FlowJo. I have no control over that.
Right.
So what they do is entirely up to them. I'm hoping, idealistically and perhaps stupidly, they are letting FlowJo do an auto comp and rework it. But we don't know that. We don't know what happens to the data once
Right.
it leaves our core. So
But
we're kind of screwed in that regard.
Exactly. But you did mention that there are, like guidelines for journals to check images, right, to see if they have been manipulated or not, right? Shouldn't we follow the same principles in this case?
Would advocate journals need to have at least a flow or microscopy reviewer, add a third reviewer.
Right.
Well, that's been talked about for the last 14, 15 years, but it still hasn't come to par.
Right.
I mean, I develop a mild Tourette's every time I open a journal of immunology. It's like, oh God, I don't believe how that got published.
Right. Anyone want to chime in?
So it's very easy to manipulate images even without Photoshop. You can do it in Fiji. You can do everything you want, and those are legit, softwares that people can use. So I think the most important thing is to always use your raw data, right? So yes, I can change things on the instrument, and that's where the training comes in, and that's where we try to help, get things so that the data is recorded correctly. But always use the raw data, and this is where I also think with image analysis and even with flow cytometry, the computational aspect, so the computational image analysis comes out, because a lot of people can see things better. Okay, so I change the contrast, I increase things, so the yellow co-localization pops out, these types of things. And that's for me, or it is for me now. It's for the PI, right? So they could see it, maybe the journal. But the raw data doesn't lie. And the computer can see what you can't see, right? The computer sees the numbers. The computer sees the transitions between light and dark. And so if you do computational analysis with the computer, then with the raw data, you're going to be a lot safer.
Mm-hmm. Yeah, I agree.
I'd say I pretty regularly encourage people, because same thing on every single image that you can pull out of an image stream, you can adjust that image display property to a dim cell, because the bright ones blow the screen out. So you turn it down so you can see the definition. And that makes then if you apply it across the board, the dim ones you can't see anymore. And so I always talk to people about, if you're going to adjust this, you have to disclose it. You have to tell them, especially if you're connecting it to a dot plot and you're saying this is the bright one and this is the dim one, but then the person looks at the images and they're like, but they look the same. And you have to explain what happened. So it's just all about that, being upfront and honest and saying what you did, because the data is still there.
Mm-hmm.
I always tell them foundationally, you are not changing what you collected by adjusting those image displays, but you do need to share with people what you did. Yeah.
So I think this particular issue it's like the abortion argument. You have 2 points of view, but at the end of the day, this particular issue you can break down quite straightforward. Is that one, if you have quote unquote compensation issues, there is something wrong in the mathematical model that you're applying to the data. I mean, your single stain controls are in some way not correct for the data you're analyzing. There's no way to get around it. The question then becomes, how accurate do you need your experiment? Then you get into, then that's de facto. Now, whether you should change it or not, that's human psychology and human perspective. You only need your experiment to be as accurate and sensitive enough to get the level of result you're trying to report. If I'm trying to have a scale and I'm trying to report the difference between a 10-pound rock and a 1-pound rock, I don't need something that's accurate to the nanogram. I can just get a less accurate measurement. And in some way, if I adjust it by 1/10 of a pound, it's not going to change the biological result that I want. So if I'm looking at an experiment where I'm trying to look at my result that I'm reporting is the percentage of a population who is 4 decades brighter than my negative, and my negative is tilted on some sort of stupid angle, and kind of like adjusting the contrast, I want to bring out the result that I want to get. I don't want to have to answer 500,000 questions that are irrelevant to the result that I'm reporting, because there's no way a compensation error screwed up 4 decades of brightness. I have the same population. So you make a human psychology decision of, do I want to spend 10,000 hours arguing with reviewers and colleagues and everything, all pointing out that my negative population has a tail to the left and explain the entire math of compensation to them? Or do I want to correct it and focus on the biological result, which is still accurate and unchanged based on the other one? Is that the right thing to do? The wrong thing to do? The expedient thing to do? In a perfect world, we wouldn't do it. It's a judgment call, I think, based on what you're doing. As long as you're sure that it's not changing the results, just becomes human psychology decision. Right. Do you tell your little white lies every once in a while?
Again, to that point, that image that David showed where you had the Western blot where they just, removed a little bit of that very, very dim band, that would be exactly the case that you're saying.
No, because that dim band was there. It was more like if they took the bright band and the bright band was on a 45-degree angle and they shifted the bright band back.
Right, but you have
about something that doesn't.
No, right.
The biology.
Right, which was the case that he showed there, where it doesn't change the understanding or the conclusions. It was just dimming that band exactly to avoid those kind of questions that then you have to, well, what is this here? Well, it's nothing.
Yeah.
The conclusions are the same.
So if I could just say, what I would say about that is that's where the documentation comes in. So if you're going to do that, which I'm not, I'd have to see it, that'd be a case by case basis for me. If you're going to do that, then you need to put in your methods or the figure legend what you did. And then you need, and this is where I think the raw data comes in, whether it's shared as part of your grant submission, grant part, NIH sharing, or shared in the supplemental data, then you should also put the raw data with your paper. So that way you allow people to go back and look at that, because so a lot of people will look at your data for other reasons, right? They might look at for a paper they're doing, and then now we don't know if that tail was compensation. You can say that it is, but I don't know that it is. So you should give the people the opportunity to then look at it themselves by putting the raw data next to it, and then documenting very carefully what you have done.
And I just wanted to agree with you as far as it ultimately has to come down to, the onus has to fall on the person generating the data. I know that as a tool maker, BD Biosciences, we struggled with the idea of, having a reference library of controls for that very reason, that it's not best practice if broadly applied, but it's nice to have and will save my butt on a bad day of controls. And the same thing for manual compensation. Being able to adjust that post facto is going to save you in cases, but ultimately, if broadly applied, it's just flat out bad practice.
Yeah, we have a question from the audience.
Yeah, I wanted to ask about the minimum number of events. I've done some imaging cytometer with rare populations. We're able to get about 100 events, and then we're like, there's not enough because traditional flow cytometer, you're looking for thousands of events. I'm also asking, as we start multiplexing these rare populations, we might be able to upfront acquire a lot of these rare populations, but once we separate the samples, we're getting less of those events. What would you recommend be that minimal number of events that you're getting for microscopy, imaging, and all the other technologies?
Anybody answers this question, I'm kicking them off the table. There is no way to answer that question right now from this table. It is impossible. So the statistics literature is full of exactly how you answer this question. This has nothing to do with flow cytometry, and it has to do with the magnitude of the changes you're trying to detect, the variance of the changes you're trying to detect, and what kind of statistical significance you want to have. And there are just, and without answering that, you can't say, I mean, you could say 1 billion, you'll probably be fine with 1 billion no matter what. But if you want to know the answer to that, you unfortunately have to do a bit of work and understand your experiment and trying to understand the sensitivity and the accuracy and the confidence intervals you're trying to get and all of that stuff. And you'll get a very straight, it's not hard. It's all been there for 200 years. There's websites you can program this stuff into, but you do have to go in and do it for your experiment.
So one of the slides that I skipped had a line that said power analysis is not only for mice. So you can do power analysis and other types of analysis like that on any type of sample. If you have a sample variance that you think you have from from your preliminary data, things like that, just like you said, you can apply that. So yeah, everyone uses power analysis to minimize the number of mice they use or rats or whatever they're using, but you can use it for anything. We use it for imaging experiments, number of cells, all of that.
I'll risk being thrown off the table, Dave. I would say, is it reproducible? Sight unseen, since I've only met you like, a few minutes ago, I'd say reproducibility. Can you do it 3 to 5 times with 3 to 5 times on 3 to 5 separate days? And do you see the same thing? That would lend to reproducibility and the fact that you might be looking at something real. And then do you have downstream experiments to support that conclusion?
Because I mean, on one day you might see it in 50 cells, another day 100 cells, and like, is it what percentage of your total? And like, we've done these on the image stream where we have like a plot that we'll put up sometimes, and you just see the more events that you get within your population of interest, the tighter those RD values get. But I mean, 1,000 is definitely better than 10, right? So the more that you can do, but if you have very rare events, and if I only find 1 in a million, but it's a circulating tumor cell, and I can verify that it's a circulating tumor cell because I have that one image, then is that statistically meaningful for that patient that still has that circulating tumor cell? Yeah. Right. So it kind of goes back to that. What is it you're trying to show? Are you trying to show shifts in populations? You're just trying to show in the presence of a few, but obviously more is always better. But in our experience, if you have verifiable 500, 1,000 verifiable real events, your RD values are always going to get better, but does become a certain point in time where you go, how much better do I need to get? Right. Is 10,000 going to be that much different than, 5,000 or 1,000? And that's where the math comes in. You have to look at your experiment and say, what's important in this case.
Okay, so everyone is getting hangry, so I think we just we'll go with one last question. So I guess it's also a low-hanging fruit here. So let's discuss the role, and we'll start with the role of core facilities, and then we'll jump into the role of companies. What roles can they play in really helping people or helping researchers improve their rigor and reproducibility? There was actually one question online, talking about, we have these researchers, these PIs that are just too busy to learn. They don't want to be educated. How can we help them actually improve their science? And maybe we could start with Travis, David.
Sure. Okay. The distant PI. Yeah. So it's a way that we can't do anything about that. I'll be honest. Like, unless they're going to come in and be trained in our core with their students, which we are happy to do. They don't charge them to sit in and watch. Even touch the microscope is really hard. So what we do is we really encourage people, especially new trainees that have never come to the core, but also over more than one time, to have a consultation with us to talk about their project, because the distant PI doesn't really want to give the information they need, and that'll help them. And then we encourage the PI to come as well. We can't mandate it, but if we've had really good success when the PI and the trainee comes, we talk about everything. We can come to some conclusion. The PI learns, we learn what they want, the trainee learns what they need, and then we kind of make a plan going forward. That also mediates things like how much this is going to cost, things like that. So that's the PI thing too. So that really helps. and that really helps a lot for the PI. Things that we can do for rigor is, I will tell you, I thought this before. The reason I'm a core director is because I've used cores my entire life. I've been very rarely where the instrument was in the lab I was in. Core facilities are instrumental to rigor because they centralize the instruments. We can keep the instruments running correctly. We can QC them correctly. And the biggest thing is if each department has its own microscope, we don't know what that microscope or instrument is like. But the biggest part, even if they're maintained well, is that what is the training like? Do you want the training to come from the horse's mouth, or do you want it to come through a telephone game of trainees that have left over the past 20 years? That is where the core facilities come in, I think, the most, is that, yes, we can buy better equipment because we pool money, but the training aspect is instrumental because we will train the people the same time every time. And then we update the training as it's needed, as the instrument evolves, as we get new ones. And that'll never happen in a department. Or I'll be honest, because I've run a flow cytometer in a lab before, a lab environment, it's very hard to postdoc leaves by 2 weeks, then what, right? So we try to keep it consistent.
Thank you.
To kind of underscore what he said in 2 words, education and training. And if you got that distant PI who's just not there, I usually will tell people things 3 times. If they don't get it by the 3rd time, it's called don't cite the core. You know, you can only go so far. It's I won't say it's an unwinnable discussion, but you have to have it. But at some point, you're going to run into a wall.
Mm-hmm. So, Robert, do you want to give some perspective on?
I mean, yeah, obviously the education and training part, we spend a lot of time training cores and individuals as well. But I think from our side, from the instrumentation and the software, it's about producing tools that aren't hiding the things, right? I mean, you know, like there are things that we'll do all the time and I'll be like, well, that's weird. You know, what's going on with that? And when we dig into it, we'll find some sort of artifact, something that's happening, some image analysis thing, or even, you know, from the spectral side, you know, unmixing it, whatever, like different things that are happening, but not just being like, whoa, let's not tell anybody about that, you know, but being willing to figure it out and then make people aware of it so that they aren't publishing things that we know internally are artifacts.
Right.
And that's on us to make sure, because obviously we're all competing, we're trying to sell instruments and everything, but in the end, the hope is that we're scientists first and we want good data to be coming off our instruments because that's what's going to help.
Right, but how do we change that? Because you know, usually companies will say that, well, this is IP, it's patented, we don't want to disclose this. How do we, and like you said, right, with all these unmixing algorithms, it's so important not to have a black box. How do we overcome that? I know it's a difficult question, but
Yeah, it is, right? Because I mean, I'm just the guy out here being like, hey guys, so I learned this thing the other week. I mean, you know, like, and it's a balance, right? And there is internal discussions about, all right, can we solve this? How do we make sure people are aware?
Mm-hmm.
I mean, you know, if you guys want to add anything to that, like, I don't want to be the only one up here being like, this is what Cytek's trying to do. You know?
Yeah, it's a slippery slope, right?
Yeah.
It's like, you know, the competitive environment is such that we have to try and differentiate ourselves from a marketing perspective. So we were trying to say ours is better than theirs, and theirs is not as good as ours, but we're not going to tell you how ours is going to do, otherwise they're going to copy it. And so it's a loop of,
right.
you know, but as far as the rigor goes, what we've tried to do is kind of echo the training and education piece by having more guided workflows that reflect just good practice, right? And obviously the data that I show, I try and demonstrate good practice, and it's reflected in the software as well. But that's why it's also really important that we work closely with the cores so that we can provide the tools they need to do all the training, because that's where the magic happens, is when you have these new users coming through. And I don't care if you have a PI who's checked out or not, the people doing the science are the ones you guys have access to. So that's where the training and education has to take place. Yeah.
Very good, very good. So again, let's thank the speakers here in the panel. Thank you very much.